Hey,
Hey, Shane.
how's it going?
Doing fine. Sorry, I wasn't sure which link. That's why I asked you. But
Mhm. Yeah,
now I know.
if you sign up with Calendly, they give you they generate, but yeah,
Okay.
it's new.
I've just been doing different things like the World Section Bank and yeah, stuff like that.
Um, have you reviewed the Jack Westin content?
Yeah, I'm almost done with it. I was going over the Chem Phys stuff from the videos with like one of your other students, and I'm up to like question 92 or something. But there's two questions I wanted to start with today from that diagnostic that you didn't go over with her. So yeah, we could start with that. Let me just start recording
Yep.
first. I sent you a request. Okay, right there. Okay. Share screen. Do you see it?
hmm
Okay, so this is the first question.
Oh yeah. So this one is not very clear in because what they're so there's something, yeah. So there's regular flow. So here we go. So like let's say this is a pipe and these are the flow like lines of like the fluid going through the pipe.
Yeah.
It's kind of like flat, and this is called laminar flow. And
Mhm.
it's named that because it's just like these streamlines. And this is how an ideal fluid is going to behave. Um, and what's an ideal fluid? An ideal fluid is a fluid which has no viscosity.
Right.
What's viscosity?
Uh, it kind of holds the fluid back. I can't give you a proper definition of it. I think of it as kind of like friction, but like not really.
Yeah. So if you imagine like each of these lines to be like a layer of a fluid, the layer the there if there's friction between the layers of the fluid, that's going to cause viscosity, which is this kind of resistance to this streamlined flow. So an ideal fluid will have no viscosity, so it's going to have this type of laminar flow in which the streamlines are equal throughout the entire fluid. And you can use the continuity equation for this. You can use Bernoulli's equation. Now, if you have a real fluid, which means it will have viscosity, there will be a flow that is of this shape. And that's because if you think about it at the center, like dead center of the pipe, let's say, uh,
Mhm.
there's just like the two kind of layers flanking it. So, and then as you get closer to the walls of the pipe, there's friction between the fluid and the pipe. So the flow has this bowed shape if it's a real fluid because of that viscosity. And this is now called Poiseuille flow. And Poiseuille is just, or Poiseuille's law is essentially kind of like Bernoulli's equation, but apply that to a real fluid.
So okay, is laminar. Ideal is laminar.
Uh-huh.
We
And
use Bernoulli's for that P plus one half pH, whatever that equation. And then you have non-ideal flow, non-ideal, which is also Poiseuille flow, whatever it is. And for that, that's like the pi r to the fourth over 8 and now.
Yeah, yeah. Now the reason that this question's kind of whack is that it doesn't, there's nothing in it that specifies what type of flow this is.
Hmm. Okay.
So, like, there's nothing here that tells us that it's Poiseuille flow. Um, so, so yeah, if we're relating the flow rate, by the way, this flow rate Q, what unit would it have?
Um, well, usually I would think that Q is A times V, that equation.
Mhm. Yep.
Um, so I mean, that's area, surface area times velocity. Uh, right.
Mhm.
So it would be meter squared times meter over second.
Mhm. And then,
Meters to the third power over second.
uh-huh. And so what is that meters cubed part? Uh,
Uh, I mean, that's volume.
yep. So yeah, so this is volumetric flow. It's
Mhm.
the rate at which a volume of something, flows per unit time. So a lot of times like so the laminar flow, like the most typical equation would be like this continuity equation, right?
Yeah.
And something that they like to ask is like, okay, what if we doubled the diameter? What would happen to the flow? Or rather, sorry. Well, actually, yeah. Let's say we doubled the diameter. What effect would that have on the flow, the volumetric flow?
Well, the thing is, that's what I thought this was the question was referring to, and that's how I did it.
Mhm.
I thought, I mean, with area, surface area, if it's like a cylinder, it's pi r squared.
Wait, so uh so we know that area is going to be like a distance squared, right?
Okay.
Right. So if you doubled, let's say, the diameter, the area would go up by a factor of what?
Two.
So let's see, right? So okay, let's say the diameter is 2 meters, right? The area would need to have what unit?
Okay, the area, I'm squared.
Good. So
Quadruple.
yeah, good. So that's the relationship between area and like a length, right?
Okay.
Um, so yeah, so here they're saying that the Q flow rate goes down by a factor of 16. So if it was related to area, then yeah, 1/4 would work, right? Because 4 squared would be the 16. Um, but there's nothing here that tells you that it's Poiseuille flow, but that's the only reason why this one, because if you use Poiseuille flow, that's the equation it's the only equation that has such a high value for the radius, like for the exponent. So we know that it's going to be related to the fourth power, the radius to the fourth power. So we essentially just have to do the fourth root of 16 just for this part. So yeah, this question, there really isn't anything in it that would help us know that it's Poiseuille flow, so it's not a very good question. But you know, you have the right answer if it was just regular like laminar flow. And then it would be this if it was Poiseuille flow.
Okay. I'm still a little confused, not gonna
So tell me,
lie. I mean, pi. So, 1 second.
Mhm. Yeah, because for this, for the laminar flow, Q is going to be proportional to like the distance or the radius. I guess I'll use radius squared, right?
Yeah.
But for Poiseuille flow, Q would be proportional to the radius cubed.
Oh, okay.
So, so you have the right answer in if it was laminar flow, but and there's nothing here that tells us that it's Poiseuille flow. So that's why it's not that good of a question. But if it is Poiseuille flow, then we would have to use this relationship.
Okay, but so let's say we have the first radius. Radius one is r to the fourth power, right?
You're saying for
So, so look, let's say that like let's say radius one um is 2.
Mhm.
Okay. So now the Q1 is 2 to the fourth power. Is that correct?
So,
Not literally, but like,
uh-huh.
you know?
So you're saying if radius one is, let's say, 2 meters, and then you said a Q value.
The Q would be proportional to 16. 2 to the fourth power.
Oh, okay. So you're doing it for the Basile.
Like
So,
what I'm saying is, can we try to get this just through substitution instead of like this like abstract, like using the variable, you know? Because that's how I usually do the problems, honestly.
um, let's see. I mean, like with plugging stuff in.
Yeah, I just like I substitute some sort of value.
Mhm. Yeah, you could do that. So, so we can do this. So like from here, so like let's say that R1 is 2 meters, and let's say R2 is something like, or rather, let's say that we don't have R2, like this question doesn't give us R2. And we can have kind of two paths here. So if we have an ideal fluid versus a real fluid, if it's an ideal fluid, our Q is going to be proportional to R1 squared because it's proportional to the area here. And your real fluid, aka Poiseuille flow, would have this to that fourth power.
Okay.
And so I'll use green. So this comes out to, you know, a value of 4 meters squared, whereas this comes out to a value of 4 times 8 times 2 again, which is 8 times 2 again, which is 16 square meters. So we can see that there is there's more of of an effect that radius has when we're dealing with real fluids than if we were simply dealing with ideal fluids.
Okay, but so for R2, the Q would then be 1 meter squared, no?
Um, let's see. So for R2, what okay. So for them, they're saying that R2 is when Q is equal to, or let's say Q1, or rather, let's say Q2 is equal to Q1 over 16, right?
Okay.
Because they're not giving us any value of R2, right? They're just saying that this new value of R2 results in that original flow rate Q being divided by 16.
Right, but if hypothetically the Q of R1 is proportional to R to the fourth, which is 16 meters squared, then hypothetically Q2 would be proportional to 1 meter squared.
You said when if Q,
So like Q2 equals Q1 over 16,
yeah,
and Q is proportional to R to the fourth. Q is proportional to 16 meters squared, Q1. So then if we plug in Q1, which is 16, technically,
okay.
Q2 is 16 over 16, which equals 1. So
Mhm.
Q2 is proportional to 1 meter squared.
Hmm.
And so R1 is 2, and R2 is 1. And so that gives us B, which is 1/2.
Okay.
Whatever. Let's just move on. I don't want to waste time.
Sure, sure.
This is like yeah.
Yeah. So,
Just I'll
just
look I'll look over later. I don't want to waste time.
yeah, sure, sure.
Okay, next. This is my beloved Doppler effect, which I get wrong every time.
Okay.
So yeah, let's go over this again. I mean, we always go over Doppler effect, so
So, yeah,
yeah.
there's this nasty formula that it could make things more confusing, but we want to see like let's have this approach to Doppler questions, right? Let's see if we can conceptually figure out if the observed frequency is higher than the actual frequency or lower than the actual frequency. Okay. Now, if the distance between the source and the observer goes down, meaning they get closer,
Mhm.
that will result in an observed frequency that is larger than the actual frequency.
Yep.
Now, if the distance between the source and the observer increases, that will result in an observed frequency that is smaller than the actual frequency.
Mhm.
So now we're being given the actual frequency, 300 hertz, and the ambulance moves toward the stationary observer. So the distance between the observer and the source, is that getting smaller or larger?
Smaller,
Now, if it gets smaller, the observed frequency will be,
higher.
yep. So we can get rid of these guys.
Okay, bingo, yeah. All right.
Now, yeah. So that can be a way of doing it. The formula can be pretty confusing, just because like the top part is like adding
Yep.
and the bottom part's subtracting. But
I
ultimately,
mean,
think about it. Yep.
most questions on the MCAT, I feel like never ask you to actually calculate it. Like they want you to understand it conceptually more than anything.
Hmm.
So, okay. All right, next question.
What if the speed was zero?
Oh, it'll be the same frequency.
Yeah. Good.
Some stuff in the past. Yeah. So this, since we're on the topic still, it's another one.
All right. Let's see.
Try read it.
Yep.
A car drives at a constant speed toward the wall of a canyon while sounding its horn at a particular frequency. The sound waves from the horn reflect from the canyon wall and return to the car as it continues moving toward the wall. What happens to the period of the reflected waves observed by the driver of the car?
All right. So let's see if we can visualize it. So a car drives at a constant speed toward the wall of some canyon. While it sounds a horn at some frequency, the sound waves reflect from the canyon wall and will return to the car as it continues moving towards the wall. All right. Um, so what happens to the period of the reflected waves? So we're only considering the reflected waves um, observed by the driver of the car.
Okay.
Huh, so yeah, how would you approach this?
Um, well,
Let's not think about let's not think about period right now. Let's start with the Doppler questions with figuring out what could happen to that observed frequency.
okay. Well, the car would the wall of like, the canyon, I guess, be the observer here?
Um, it'll it'll
Like
be
technically.
um yeah, you can think about that, right?
Okay. Well, anyways, as it gets closer, um, I mean, I'm half cheating because still kind of remember the explanation for this, but as it gets closer, the frequency
Hmm.
gets higher
Good.
because yeah, it gets higher. Like the
Hmm.
okay, yeah,
Good, good, good. Now we're looking for a frequency getting higher. Now let's think about then period. What's period?
I mean, period is inverse to the frequency
Okay. So we should see period go up,
down.
down. So maybe we can get rid of these,
Yeah.
right? So now we have C, the period of the reflected waves will decrease because the observer moves in the opposite direction from the waves, or the period of the reflected waves will decrease because the speed, well, what do you think about these choices?
Well, see, the thing is, I don't think speed can increase or decrease, right?
Because it's the speed of the wave, right?
Yeah, yeah.
So the speed of the wave, like in this case, a sound wave, right? That's not changing, right? You can't really change that with Doppler. You can change the speed of the car. In this question, I guess you don't change that because it's a constant speed or whatever. But the speed of the waves are just going to be the speed of sound in air, right?
Mhm.
So is the answer C?
Yes, I'm pretty sure it is. I
Mhm.
mean, I'm just trying to reason it through, but
Yeah.
like, I still don't fully understand C either. I mean, I kind of do, but it's like, right, if the car is moving toward the canyon, they reflect off and they're going in the opposite direction. But how does that connect, like, to the period? I mean, because
So I think that the wall, yeah, so the observer I think is going to be the same as the whatever, the well, I was going to say same as the observer, just because it would bounce back. But yeah, what were you thinking?
I mean, I wasn't thinking anything. This is just more of a process of elimination sort of thing, I guess.
Mhm. So I think the car is also the observer. So I think that like when you first said that we can think of the mountain or whatever that thing is, as the observer, I was like, yeah, um, but technically it just bounces off the wall and then it comes back to the driver. So the
Okay.
source of the sound is also the observer. So I think that's what made it so that choice C was one that you didn't pick because it said something about opposite directions. Yeah.
Yeah.
So yeah, at first like I thought it was fine to think that the wall is the observer, but I guess it's not because it would make C not true. But yeah, it goes back. It's kind of like how sonar works, right? Or like echolocation. Like a bat will shoot some sound, and if it hits a wall and it bounces back to the bat's ear, the bat can be like, oh crap, the wall is like this far from me.
Yeah.
Same thing with sonar and like a submarine or boat or something like that. Um, so yeah, the observer is the same thing is the same as the source of the sound here, but it just bounces off of the wall and then comes back. So that's why it's the opposite direction,
Okay.
right? So, but but yeah, ultimately the way that we answered this was okay. Yeah, are we going to see like we black box the period part? Let's not think about that first. Let's think about frequency first. And you were able to determine that the frequency of the observed frequency should increase. And you also know that period and frequency are inversely proportional. So if the observed frequency should increase, the period of the reflected waves should decrease. And that gets rid of A and B. And then C, like even if you weren't unsure about this part, you should be sure that this part is not changing because we're that's the speed of the actual sound wave itself, right?
Yeah. Yeah. Okay.
Yeah.
This is the last one. That is like about Doppler effect. An apparatus consists of a cart that moves along a horizontal track inside a sealed tube that can be filled with various gases. Researchers attached a speaker to the cart, and a sensor that can measure sound is located at one end of the tube. Which of the following procedures will allow the researcher to change the speed of the sound created by the speaker?
All right. So now this time we're changing the speed of sound. Okay. So horizontal track inside a sealed tube, various gases, and that matters because sound travels at different speeds depending on the density of gases. Now, meaning more dense, faster. Researchers attached a speaker to the cart, and a sensor that can measure sound is located at one end of the tube. Which of the following procedures will allow the researchers to change the speed of sound? Okay. So, I mean, okay, let's see. So this is exactly what I was kind of thinking of. Um, with the various gases, the denser the gas, the faster it'll travel. So my guesses here would be C.
Mm-hmm. Yep, yep.
So
I didn't know that. I mean, is this kind of like referring to the fact that like sound travels fastest in like solids, then liquids and gases?
exactly, because
Okay.
sound is the vibrations of actual things. So like, yeah, so if you have so like why is it faster in a solid? Because it's denser. So you can just apply that like knowledge that you have that it's faster in solids, then it gets slower in liquids and then slowest in gases. You could just take that knowledge that you already have and then use that to think, hey, what's another way of saying solids? Okay, it's just more dense. So the more dense the medium is, the faster it'll travel through it. And it's the opposite of like Schnell's law with n equals like the index of refraction for light.
Yeah.
That's the opposite because the denser that gets, the slower light travels. So light opposite, but with sound, it's the velocity of sound through a medium is proportional to the density of that medium.
Okay. Sound is a pressurized wave, right?
Mm-hmm. Yeah, it's uh, pressure waves. Yeah.
Okay.
Mm-hmm.
Why did they say speed is independent of frequency? V equals F times wavelength.
Uh-huh.
So why is it independent?
Oh, because I mean, if you have a higher pitched sound, uh, it's not gonna really make it go faster or slower.
It's going to change the wavelength?
Uh, yeah, it can change the wavelength. I mean, if we're dealing with light frequency, we don't change. That's kind of like a thing. But
Okay.
but 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 yeah, the wavelength will inversely change as frequency changes. But yeah, the speed of the sound is the thing that doesn't change unless you change that the density of that medium. Um, so yeah,
Mm-hmm.
think about that in real life. Like if it's a high pitched sound, does it mean that it's moving faster? Doesn't mean it's moving faster.
Okay. Yeah, we'll do these then next. Um, I've always just not in terms of chem, like my worst topic is acids, bases, buffers, titrations, all that. So, I mean, I think this is a pretty simple question, but I just kind of forgot how a buffer works.
Yep. So buffers are going to resist changes in pH. And
Yep.
that will typically consist of a weak acid and its conjugate base or a weak base and its conjugate acid. So we want to look for weak things here. Now,
Okay.
anything jump out at you if you're looking for strong things, perhaps?
NaOH is
Yep,
a strong base.
super strong. So that's not going to be it.
What does NaCl classify as? A compound that dissolves in water into ions?
Yep, it'll break its table salt. It just will break apart into Na plus, Cl minus.
Okay. How about NaBr and NaCN?
Na,
CN minus?
yeah. So that Cn minus is could be so the Na plus whatever is gonna not do anything, but the Br minus, that halide, is going to be quite reactive. Um, because it has very high electronegativity, right? Um, but the NaCN, so okay, so let's say we had hydrogen cyanide, like yeah, that can break apart into H plus and Cn minus, and that is well actually that's a weak acid. Um, let's see. Oh, you know what? But this is from like HBr. Like HBr would break into H plus and Br minus. So this is a conjugate um, base of a strong acid here. So
Okay.
that won't work um, because we have the conjugate base of a weak acid, HCN, and we have the conjugate base of a strong acid, the NADR. So that won't work. And then nitric acid here is a strong acid. And this is the conjugate base of that strong acid. So that's out. And this is acetic acid, which is, what is
Weak acid.
Mhm. It's like vinegar, right? So yeah, usually like, and it's like an organic acid, like it's a carboxylic acid, right? And usually those are not going to be very strong acids or anything like that. Compared to something like HBr, HCl, HBr or HCl or something like that. So, so what we're looking for here are buffers, which are just going to involve, it has to be something weak. It'll be a weak acid in its conjugate base, a weak base in its conjugate acid, cannot be anything strong.
But is it the conjugate like base of a weak acid? Is it also a weak base or is it a strong base?
Um, well, typically, uh, it'll be a weak conjugate because it'll want to be the thing that it breaks into in order for something to like move. Like, so for instance, like here, right? Um, where HBr is a strong acid and Br minus is its conjugate base. Now, if Br minus was a strong conjugate base, it would want to get protonated. And if it wants to get protonated, it would go backwards. But that would contradict the fact that HBr is a strong acid because we define a strong acid as it's going to be 100% dissociated,
Okay.
right? So strong acid must have a weak conjugate base. Weak acid must have strong base must have a weak conjugate base because it's gonna just do one or the other. Whereas weak ones, right? A weak acid, HCN, a weak conjugate base, CN minus, that would go like this. And it'll basically just be this kind of cycle where you don't have any definitive change in anything, meaning like your pH won't change much.
The strong base is conjugate weak acid.
Yeah, so I would say like okay, strong means 100% dissociated, which prevents the reverse reaction. Okay. If we have HCl and that dissociates into H plus and Cl minus, right? From the amount of H plus, we can calculate, we can figure out its pH, right? Now, the Cl minus is its conjugate base. If it was a strong base, what do bases like to do? They like to get protonated. So if it was a strong base, this should just go backwards to make HCl.
Okay.
If it went backwards to make HCl, guess what? We're using up H plus. So we're going to have less H plus, our pH is going to go up.
Hmm.
But if it's a strong acid, it's defined as not going backwards, which means that that conjugate base must be weak.
Yeah.
Mhm.
How do you know that like NaNO3 is the conjugate base of HNO3?
Oh,
Like, doesn't H plus has the same charge as Na plus?
yeah. But they might have the same charge, but the H plus is what we must use to determine pH, right? Like that's the thing that like an acid would be defined as donating, and that's what a base would be defined as accepting, right?
Okay.
Um, but when you see it with like the Na, it's really just like all of these things with NAs just mean that that's like the salt form of it that once it enters water will immediately dissociate, and that sodium ion will dissociate and not do anything. It'll be a spectator ion. But the NO3 minus is the conjugate base of the HNO3 because the HNO3 is an acid, which means it'll get deprotonated. So it'll break apart into H plus and NO3 minus.
Mhm.
And this NO3 minus is the same as NaNO3 because that would just dissociate into the NO3 minus. But it won't dissociate into anything with a plus because NaNO3 doesn't have an H plus. So
Okay. So CH3COONA is a weak acid, a weak base.
weak acid.
The first one is a weak acid. The second one is a weak base, right?
Mhm.
Okay. Next is this. So how do you know that sebacic acid or whatever it is called? How do you know right away that this is a weak acid?
Uh, okay, let's see. Well, it's a organic acid, which tend to be weak, but let me see what else I can see. Dicarboxylic acid found in urine, a 200 milligram sample of sebacic or whatever acid crystals, oh, okay, would have the highest solubility in dilute aqueous. Okay, yeah, we probably don't even need to, well, we don't need to necessarily know that this is a strong acid or a weak acid, but we know it's an acid. Um, and an acid is defined, as we use a Bronsted-Lowry definition. So an acid is defined as a proton donor. So what would what if we take this acid and we put it in, like what can we put it in for it to get deprotonated the most, right? So for instance, if we just put it in water, it'll get deprotonated a certain amount, right? Um, and if it's a strong acid or whatever, it'll get maximum deprotonated 100%. But if it's not 100% getting dissociated, what can you put it in to make it get dissociated more? Well, you could put it in a base
Mhm.
because, if you put it in water, it'll get dissociated and into, H plus. Um, and if it's strong, it'll get dissociated all the way. But if it's not strong, it won't get dissociated all the way. So what can you do to make it get dissociated all the way? Well, you can add a base to it. If you add a base to it, a base is defined as um, being a proton acceptor, and whatever protons have not dissociated yet from the weak acid, you can force it to get deprotonated by adding a base. So for this question, we don't even need to know that whether this is a strong acid or a weak acid, because even if it was a strong acid, the highest solubility would still be in a base, a weak base.
All right, next is this.
Yeah.
So I mean, I know this has to do with, like the pi equals IMRT equation. That's all I really
Mhm.
know.
Yeah. Now, what's the I represent?
How many ions it dissolves into?
Uh-huh. So HCl would have an I value of what?
Um, one. I don't
So
know. Does H plus count? That's I guess it does, right? Two.
two, right? Because this is how you should think about it, right? Let's say we have HCl, right? Let's say we have 1 molar HCl. It'll dissociate into 1 molar H plus and 1 molar Cl minus for a total of 2 molar of stuff,
Mm-hmm.
right?
Yeah.
So same thing with, let's say, CaCl3. Well, that doesn't even work. CaCl2. What would the Van't Hoff factor be for the I value be for this?
Uh,
So think about how many things it associates into.
I mean, I guess three.
Yep. So now what about glucose?
One.
Good.
Yeah.
That's the Van Hoff factor. That's what that means. Um, and so yeah, you'll have your this guy, right?
Mm-hmm.
And so we start off with, let's say, M1, I'll call that, like the molarity, capital M, for one. Um, pi one, I'll call this 1.9 ATM. And then they're asking us to find pi two. Um, approximated as zero point 0.1 HCl. Wait, what? Wait, sorry. Osmotic pressure of this, zero point 0.1 HCl under the same conditions. 0.1, wait, where is that? Okay, so I guess this is M2, right?
Yeah.
Okay. Now, thankfully, we're keeping like, because technically there would be like one, like, and then for two and two, which really just means that we don't have to worry about the things that are constant, like the RT.
Uh-huh.
So we can probably just make a proportion here, right? We can say um pi, let's see, 2 over pi 1 is equal to m 2 over m1, and we just have to solve for pi 2. So pi 2 is pi 1 m2 over m1, and then this would be the 1.9 ATM times the M2.1 molar divided by M1.04 molar, right?
Yeah.
And let's see. I can do 0.04 or
Can we just estimate 1.9 to be 2?
mhm. So let's see. This would be 5, right? So I,
Okay.
I 1.9, I turned that into 2, and then I multiplied it by
Yep, yep. It's 5.
0.1, and then 0.04 goes into 0.25 times. Now that's an we rounded up, so our answer is going to be a little bit less than 5. So
Hmm.
would that be D?
Yeah, yeah.
So it seems like we didn't have to even do any Van't Hoff stuff here. And the reason we didn't have to do anything with Van't Hoff is because that hasn't changed. That's the reason.
Hmm. Okay. Next is this. I don't even think we need to read the passage for this question. So basically, it says like an infrared thermometer uses a lens to focus incoming radiation onto a small sensor. Which kind of lens should be used when the sensor is 1 centimeter behind the lens?
Behind the lens. Okay. So, what are your thoughts?
First of all, focusing something is converging, so we can eliminate C and D. Because in order to focus something, right, you use a convex lens, which is converging.
Mhm.
Um, yeah. After that, I was just confused because let's say we have the sensor to 1 centimeter behind the lens. If we use the correct answer is A, but if we use the something with the focal length of 1, wouldn't there just not even be an image when it's like on the focal length of 1? Remember we discussed that?
Um, let's see. This is the sensor. Would be the object distance, image distance. Um, so okay. Longer than I think the when the image distance is, let's say, Okay, so you were saying for the um okay, what were you saying about the about this?
Okay, I said if we have a focal length of 1 centimeter, if we put something on the focal length, wouldn't there not even be an image?
Um Focal length okay, if the object is at the focal length, you're saying?
Yeah.
Okay. Now, I think they're telling us that or asking us for the like I don't know if they're so okay. I think that the distance between the lens and that sensor could be the image distance. Um, and so if the sensor, so we have then if it's going to be focal length of 1, focal length of 2, and the image is 1 centimeter, then is this going to be equal? So could the focal length be equal to the image distance, or could it be larger than the image distance? So if the focal length is mm, I mean, okay, so I guess what you're saying is that if the object is at the focal length, you're going to get no image, and that's true. But from what I can see, it doesn't seem like we have information about where the object could be placed. But I believe that the um, the location of the sensor is where the image should focus. So
Yeah.
image distance should be the 1 centimeter value. And
Hmm.
then, and yeah, if that image distance is equal to that focal length, you would get an object distance of infinity, but that would just create a tiny inverted real image. So that's if the image distance is equal to the focal length. So that would be the case for choice A. Choice B involves a focal length that is larger than the image distance, and we don't have any situation where that could be the case because the smallest that the image distance can be would be that focal length. It can't be smaller than that. I mean, the object distance can get smaller than that, and that would make that, you know, large upright virtual image, but the image distance cannot be smaller than that focal length. So I think that can be a way of solving this.
Plugging stuff into the equation, I guess.
Um, if you plug it into the equation, you would just get some type of you'd get object distance is infinity, which if it's 1 over the infinity would be like the zero. So that means like yeah, the focal length would be equal to the image. But um but yeah, I think that could be a way of doing it. I just think that the is that you can't the smallest image distance that's possible would be when the image distance is equal to that focal length. So you can't have a image distance smaller than the focal length. So I think that's why it um wouldn't be B.
That's the answer.
The answer is B.
Oh wait, it's A. Oh my God. Sorry.
I'm gonna jump out my window.
No, no, no. Okay, it's A.
Oh yeah, this is actually a definition of focal length that I didn't tell you about. Like another this is a good thing to see then. So yeah, that's what this is like another like a actual definition of focal length. It's so like we see that the parallel light rays are coming in on the left side of this lens. And when they hit the lens, they start to converge. It can also be when they diverge or whatever, but the distance between that lens and like where it converges is another way of defining the focal length. But I think the thing here is that this focal length here that kind of like directs this to be towards the sensor also should be where the image is. Let's see if you can scroll down a bit. Let me see if they talk about that. So erase what I wrote. Um, okay. Okay. So I guess the way that I solved this was just kind of using that optics table and just knowing that the image distance can't be smaller than the focal length, and I equated the focal length to the image distance here. But what you can also do is understand that a focal length could be defined as the, if it's converging like this, the distance between that converging lens and where they ultimately converge at a single point. So that's another definition of focal length. But yeah, I would, in addition to that, just say that if the image distance can be smallest when it's equal to the focal length. Um, it can be larger, like out here, theoretically, but it can't be smaller. It can't be anything here. So that's why it can't be B, because B talks about a focal length being um, uh, larger than the image distance.
So DI less than F is not allowed.
Yeah.
Okay. Okay. Next is this topic. Um, one sec. Um, let's do this first, actually.
Okay. Let's see. Autonomous retrotransposons are segments of DNA that encode specialized enzymes. These enzymes allow autonomous retrotransposons to move between different regions of a host genome via RNA intermediates using mechanisms similar to those of retroviruses. Um, Some autonomous retrotransposons are segments of retroviral DNA that have been partially inactivated by mutations. Given this, which of the following mutations would most likely convert active retroviral DNA into an autonomous retrotransposon? Okay, retrotransposon seems to be able to move to different regions of the host genome using through RNA intermediates that use mechanisms similar to that of retroviruses. So when I see retrovirus, I'm thinking of reverse transcriptase, aka turning RNA into DNA. Um, okay, and some of these things are inactivated by mutations. Which of the following mutations would convert active to or active into the autonomous thing? Okay. Okay, some of the autonomous ones are partially inactivated. Okay. A mutation that prevents the expression of functional reverse transcriptase. Um, a mutation that prevents integration of retroviral DNA into host genome. A mutation that prevents the expression of envelope proteins. A mutation that prevents RNA polymerase from transcribing retroviral DNA. Okay. What are your thoughts?
I have no idea, honestly. I
Okay,
don't know.
let's see. Okay, so it seems like they're saying that the retrotransposons are segments of DNA that can move around within the genome. Um, and they do it using RNA intermediates, which is similar to how retroviruses replicate, meaning, the RNA turns into the DNA, and I guess when it turns into the DNA, it can integrate back into the host genome. So that's retrotransposon. Autonomous ones enable them to move. Okay, wait, can move. Autonomous move between different regions using. Okay. Okay, so autonomous ones literally must use, I guess, reverse transcriptase. Cool, okay. And then they're asking convert active to or convert active retroviral DNA into autonomous transposons. Um, I'm guessing they're talking about the ones that are partially inactivated. So let's see. A prevents the expression of functional reverse transcriptase. If you do that, you're not going to be able to convert RNA into DNA, which would disable reverse transcriptase, which you would need for both the retroviral DNA as well as the um, transposon thing.
Hmm.
Um, B, integrate their DNA into the host genome. Seems like they both kind of do that as well. Let's see. Envelope proteins. Um,
So, do retroviruses have envelope proteins?
um, my understanding of a retrovirus is just any virus that um has reverse transcriptase. And I guess if we're talking about like the ones that infect like animals, they would have an envelope like like around it, uh, rather than just like the geometric like capsid. They could have the envelope with the spike proteins and stuff around it. Um, so I think what I'm so okay. I think the question stem is a little verbose, but I think what they're if I and you want to, yeah, when you see questions like this, you really want to understand. Oh,
Excellent.
yeah, you want to, I guess, understand, I guess, or simplify what it is exactly that they're asking. And
Mhm.
what I can see is just maybe if we're being asked to convert active retroviral DNA into autonomous retrotransposons, and they talk about some of these retrotransposons being segments of the retroviral DNA that's partially inactivated. What can be partially inactivated? Like, or what can distinguish active retroviral DNA into partially inactive retroviral DNA? So so A and B have things that would affect retroviral DNA as well as retrotransposons. I think C would only affect the retroviral DNA, not the retrotransposon, because the retrotransposon moves around within the DNA. Not it isn't something that necessarily can be moving from like one host organism to another. So maybe something with that. Let me see the rest. Um, Mutation that prevents the RNA polymerase from transcribing the retroviral DNA. Um, and that's just regular transcription, nothing special with reverse transcriptase. So if you mess that up, it'll mess up both the retroviral DNA and the retrotransposon. So like I'm maybe leaning on C, but I don't like this like some. So okay. Autonomous retrotransposons, segments of DNA encode specialized enzymes. These enzymes allow, okay, them to move between different regions of a host genome. Okay. Using mechanisms similar to that of retroviruses. Okay. Some of these are segments of the retroviral DNA that's inactivated. Okay. Which of the following mutations? And okay, they can get inactivated by mutations. Which mutations would convert active retroviral DNA into an autonomous expression of function? Hmm.
Yeah, it's a lot of mumbo jumbo.
um,
whole lot of mumbo jumbo.
Yeah.
What is the difference between an episome and a transposon, if there is anything? Or transmosome? I always get those confused.
Uh, what was the thing? You said episome?
Think.
Segment of DNA that can exist independently or integrate into the host genome, and that seems relevant.
Episome versus transposon.
Let's see. I think I wrote something before about copy and paste versus cut and paste, but let's see,
Mhm.
like in my old notes or whatever.
Episomes are plasmids that have integrated into the bacterium genome and are part of it. Okay. Okay, maybe I should just submit and we'll go over it.
Sure. All right, okay. Good. So, yeah. Let's see. So that's what I was trying to draw here. But like, yeah. So like all viruses have, this type of like icosahedral kind of geometric shape. Um,
Okay.
but and you get to see that in like a bacteriophage, right? Um, which is a virus that infects bacteria. Now, that's because bacteria don't have like a plasma membrane like we do. Now, so imagine that same icosahedral like bacteriophage, but now it has its own membrane. That's like the membrane of a eukaryotic cell. So that's what I was like kind of drawing here with these spike proteins and stuff. And so I was really just like, so I was like kind of, yeah, when I was reading the mumbo jumbo, it was like, it was saying, okay, it was saying that these retrotransposons can move around within the genome of that host cell.
Mhm.
And using things that retrovirus would use, which in other words means that it's using reverse transcriptase. And so the way that I like led me to C, like to lean towards C, was just I kind of simplified the question into asking what's the difference between like a retrovirus and the retrotransposon. And from what they're telling me, they're saying that the retrotransposon can move around within a host genome, but can't infect another cell, whereas a regular retrovirus can infect another cell. So I kind of answered it that way. Um, Let me see what they're writing here. So let's see. They're saying retroviruses in enveloped viruses. That's that, this thing. Upon entering a host cell, the envelope and capsid disassemble, releasing viral RNA and proteins, and then the viral reverse transcriptase converts the RNA into double-stranded DNA that's transported into the nucleus. Something called integrase facilitates that integration of viral DNA into the host genome, and then it can replicate using the host genome's machinery. Um, then it says that autonomous retrotransposons move via RNA intermediates using mechanisms similar to those of retroviruses. In general, transposons are segments of DNA that move between different areas of the genome. So that I take that to mean that it can move around within the genome of the cell, host cell that it's already infected and it's already inside, right?
Mhm.
And how can we, and it does that using mechanisms similar to those of retroviruses, but unlike a retrovirus that can infect other host cells, a retrotransposon can't do that on its own. So, um, so yeah, they give us some more information. Retrotransposon is initially transcribed into mRNA, which is then transported into the cytoplasm, and translated to produce reverse transcriptase and integrase. Reverse transcriptase produces the double-stranded DNA from the mRNA, and integrase facilitates the integration of that newly produced double-stranded DNA into the host cell of the genome. And then they say that some of these autonomous retrotransposons are segments of the retroviral DNA that have been partially deactivated by mutations. And because fully functional retroviral DNA must encode the protein components to assemble new viruses, if you have a mutation that prevents the expression of envelope proteins, means that it would stop the production of infectious retroviruses. Um, so we're essentially trying to figure out, okay, what's the main difference between a retrovirus and an autonomous retrotransposon? And I really just looked at or focused on this part. So I'm thinking of in a host cell, the retrotransposon can move around within the genome of that host cell, but it cannot infect another cell.
Mhm. Okay.
So, and now here we have a nice succinct definition. So just remember that a transposon can move around within the genome of that one cell. It can't go into, it can't infect another cell, not itself, meaning not the transposon itself.
Review what you just said?
Yep. So yeah, so we can, so essentially the way that I viewed this question was that I asked myself, what
Mhm.
can distinguish like like active retroviral DNA, which I'm equating to, retroviruses
Mhm.
from autonomous retrotransposons.
Yeah.
I know that a retrovirus can infect various different cells, and a retrotransposon can move around within the genome of a single cell, but it cannot go and infect other cells. And the reason for that is because it doesn't have that envelope. And that's what this thing is. Like, when you see the picture of like the COVID, whatever, it has those spike proteins, right? And those spike proteins and the envelope around it is what enables it to infect like a eukaryotic cell. Uh, because if it infected a bacteria, it would just be that capsid, right? It would look like this.
Mhm. Okay. So the difference between a retrovirus and a transposon is that the retrotransposon can move around the genome of a single cell but cannot go infect other cells like a retrovirus.
Correct.
Retrovirus. The reason for that is it doesn't have the envelope with the spike proteins like the retrovirus does.
Yeah. Yeah. And if you wanted to add to that, you can say a bacteriophage, right, can infect bacteria, but cannot infect eukaryotes because of the same reason, aka It does not have that envelope.
Uh,
I actually did a little bit of like some bioinformatics yesterday to update some of my code. And I compared like COVID to bat COVID and hedgehog COVID and all that stuff. The first time I did it, it was like a 69% similarity with bat COVID, but I knew that it was the closest to pangolins, but I couldn't find pangolin DNA at the time. But this time, yesterday, I found it and I did an analysis and it was like 82% similar to that.
okay. All right, next. So I want to definitely touch on electrochemical
Yep.
cells. So for this, there's like I guess two questions, so it would make sense to go through the passage quick. Yeah,
Mhm.
17 and 18. All right. Should I read it or
Um,
should you?
Let me see. I'll read it. So,
Hmm.
researchers investigated the role of an electrochemically enhanced sulfate radical. Okay. Um, you know what that means? Radicals?
Radicals have one electron.
Yeah.
Um, that's really it.
Yep.
Yeah, Ross.
Ross, yep, reactive oxygen species and antioxidants. Can't
Yeah.
forget that. Uh, okay. Um, in the inactivation of antibiotic resistance E. coli bacteria, an electrochemical cell, figure 1, was constructed using an iron electrode um, and a graphite electrode. Interesting. Um, You've seen that? Okay. Connected by a copper wire joined to a voltmeter. Both electrodes were placed in a beaker containing 3 millimolar solution of sodium sulfate and a 50 millimolar solution of peroxydisulfate salt. Okay. During the operation of the cell, ion was formed at the iron electrode. So if that is forming here, this is getting oxidized or reduced, the iron.
Wait, can you remove the 69 and the 82?
Oh, yeah. Yep.
Okay, thank you, thank you. So, um, repeat your question.
Oh, yeah. What's happening here? Oxidation or
Here it's losing electrons, which is oxidation.
yep. And yeah, they didn't tell us anyway, so yep.
Oh,
But
yes. Yep, yep.
no, and
Wait,
then
that's oxidized to Fe.
so okay, that okay. So I see. So this is what's happening to make the this is what's happening at the iron electrode. And then, I mean, I guess and then through here, it gets oxidized further into the Fe3 plus. Okay. By the reaction with that peroxydisulfate. Okay. At the graphite electrode, Fe2 plus was regenerated. So over here, the Fe3 plus seems to get reduced to regenerate the Fe2 plus. Okay. And then that's what we have here. The reactions in the cell also produced both sulfate and hydroxyl radicals, i.e. species with an unpaired electron. Sulfate radicals look like that. They inactivate E. coli through membrane destruction and were produced in the electrochemical cell by reaction 1. So I'm going to just write something like this, inversely proportional to E. coli. And we have our reaction here. And cool. Okay, whatever. Now, operation of the cell generated enough energy to cause the electrolysis. Okay. So, electrochemical cell has properties. Electrolysis is going to be an electrolytic cell, and it's going to be like opposite properties, right? Like non-spontaneous, um, stuff like that. Okay, so in which water?
So
Yep.
I have a question real quick. With electrolytic versus voltaic, which are the two common ones, both have current going from cathode to anode, and then electrons are going from anode to cathode, right?
Um, kind of. It they're so like in a voltaic, it's you're connecting something that gets oxidized to something that gets reduced. So electrons are going to go from what gets oxidized to what gets reduced. When it's an electrolytic cell, you have an outside source of electricity,
Hmm.
causes the electricity to go to like the so the initially the anode was the part that was negative in an electrochemical cell, which means that the anode gets oxidized, it loses electrons, and it goes up to the cathode. But electrolytic, you're flipping the signs because you want to recreate that charge gradient. So the anode actually gets like electrons going to it.
Okay, wait. Okay, but when the electrolytic cell is being when it's charging, when there's like a outside source, when that's happening, electrons are going from cathode to anode.
Um,
But when it's discharging, when you remove the outside source, the electrons are gonna go reverse.
so uh so they'll go from anode to cathode in an electrochemical, voltaic cell. But when you have an electrolytic cell, they're not going to be coming from anything in that cell itself. So it's not going to go from cathode to anode or anode to cathode. It's going to come from that external electric source, and it's going to go to the uh, now positively charged anode end. So
Okay. So what's gonna happen at the cathode?
uh, that's just going to be momentarily negatively charged, and that means nothing, like no electrons are going to uh, accumulate on it. So you can, I guess, well, okay. You can kind of perhaps say that it'll just be the opposite, kind of, but because I see certain diagrams where it's just that the anode gets reduced. Um, Honestly, well, here, hold on. I'm gonna just put this picture in. Honestly, it's really just what's in here, and you can see that your anode is no matter what is going to still be the site of oxidation, and the cathode, no matter what, will still be the site of reduction. It's just that for the electrolytic cell, the charges get flipped, and that's just because we are introducing that external, yeah, source of electricity.
Hmm.
So you can see for this galvanic cell on the left, the anode negatively charged, oxidation happens, cathode positively charged, reduction happens. Electrolytic cell, anode just now has a positive charge, cathode has a negative charge, but it's really and they're kind of showing, yeah, they're showing the in black. Yeah.
Gotcha.
So they see the things in black, like the things in black didn't change, but the stuff that is harder to see, yeah, like this black and that black hasn't changed, this black and this black hasn't changed. But what did change is like, and they don't really draw it, but really electrons can be coming inwards here to this now positively charged anode, and then they can go back to that negatively charged cathode. But ultimately it's not like it's not like we're like you're not going to have reduction at the anode um and oxidation at the um cathode, even though it kind of may look like that here. But you're really just getting electrons coming in from that outside source towards the positively charged anode.
Yeah.
Yeah. Okay. So let's see. All right. Electrolysis of water, in which water was oxidized at one electrode to form H plus and reduced at the other electrode to form OH minus. Consequently, some of the available OH minus reacted with the sulfate radical to form hydroxyl radicals, which also aided in the inactivation of E. coli. So hydroxyl radicals also mess with mess with E. coli. Okay, to determine which radical species predominantly inactivated E. coli, two radical scavengers, methanol and tert butanol, were used to evaluate the contributions of the OH radical and the sulfate radical. The hydroxy radical doesn't have a charge, so let me get rid of that. Okay. So we want to see, okay, what works better, right? So um, monitored E. coli concentration in the active cell with and without the radical scavengers is a function of time. So concentration of E. coli is going to be our independent variable. And the dependent will probably be, let's see, yeah, you can scroll down some. So log of C over C. Okay, concentration, initial concentration of E. coli, got it. And C is the concentration of E. coli at a specific time in the experiment. Oh, time is the independent variable. But anyway, so we see they all start at 0 and no, okay, so it seems like the tert butanol doesn't really cause any change. It's pretty much exactly the same amounts as if you don't have any radical scavengers. Um, but if you have one methanol, it seems to play quite a role in increasing that. Well, if it increases that amount and C is the concentration of E. coli, it seems like the one methanol, the methanol is a pretty lousy uh you know,
I
scavenger.
will say we don't have to talk about this that much because I got that question right. So I don't want to waste time.
Huh? Oh yeah, I don't know which question yet. I was just saying that's what I observed from here. I observed that
Yeah, yeah. No,
much.
I know. I'm letting you know. I know you didn't know, of course. Um, okay. Well, yeah, I had trouble with this one. Like, so they basically say, what change to the electrochemical cell would result in a voltmeter reading of 0, right? So the voltmeter, I get like the cell wouldn't be working.
Okay, so I guess what they could be asking here is what would happen. Okay, so like for a voltmeter reading of 0, we are gonna have no movement like to complete that circuit because we have like whatever electrons going, let's say here, and then through here to close that circuit, right? So disrupt we want to disrupt that. So what we're probably going to do is so this is the thing that seems to, oh yeah, this thing over here. That's like the salt ridge. So, um, yeah. So this voltmeter reading of 0 would mean that there's no potential difference between the electrodes. And what we so this is so in order for, because okay, so we have changing the wire shouldn't change anything, and then this replacing this with this is not changing anything because we're not dealing, because the entire thing I think that they're trying to ask us here is like, so this entire thing is set up in this way to have a closed circuit. So so for instance, Fe2 plus to Fe3 plus, it means we're losing an electron that way. And it should go opposite to the direction here. So here, this is gaining, I believe, electrons, but my guess would be then this has, I mean, the same sulfate kind of thing. So
I'm just confused about what's even going on in this electrolytic cell, like electrochemical cell.
mhm,
Like, I mean, this is getting oxidized, the FeS.
mhm.
It gets oxidized. Um, I don't get the process of like what the S2O8 2 minus, what's the point of that? And then the Na2SO4 and whatever.
Got it. So yeah, at this iron electrode on the left, we are getting the solid Fe into Fe2 plus, right? And that means that oxidation is happening at this iron electrode, which means that it's losing electrons there. So we should see electrons go, whoops, in this direction here. And so that's what's happening at that Fe electron. I'm sorry, electrode. At the graphite electrode, we have Fe3 plus gaining electrons to reform Fe2 plus. So here we're having reduction, right?
Mhm.
And that makes sense because this copper, oh, sorry, this graphite electrode is gaining electrons. So yeah, that's where the reduction is happening. And that reduction recreates this Fe2 plus, right? Now, we also see that Fe2 plus can be involved here in a reaction with the S2O8 2 minus over here. So uh okay, so during the operation of the cell, Fe2 plus was formed at the iron electrode, that's here, subsequently oxidized to Fe3 plus by a reaction with S2O8 2 minus. That's what's happening here. And so here we see that the Fe gets oxidized, which means that the S2O8 2 minus should be reduced, right?
Mhm.
Right?
Okay.
And as it gets reduced, it forms this free radical, right? The sulfate radical.
Yeah.
We know that, yeah. So that's what's kind of happening here. Um, right? So so basically iron electrode gets oxidized, loses electrons, to form Fe2 plus, and Fe2 plus can react with S2O8 2 minus to turn into Fe3 plus and the radical SO4. And at the graphite electrode, the Fe3 plus can be reduced into Fe2 plus, and that's where the reduction takes place. So oxidation here, reduction here, and we need this to like keep going. Like essentially, like electrons are coming here, and they need to go back to that essentially to complete the circuit. So typically that's done with a salt bridge, and here it's kind of done in a similar way with like this being like what separates like the sulfate here from like this I forgot what it was called, but peroxy disulfate. So that's essentially what we're doing. We're essentially creating a closed circuit loop that would give us some value for the voltmeter, which would measure the voltage differences between these two electrodes. So now they're asking, how can we change this electrochemical cell to give us a voltmeter reading of 0 volts? So how can we make it so that we disrupt this circuit, disrupt the
Mhm.
electrons from flowing through the circuit, right?
Okay.
So, so now if we look at these choices here, so what we want is we want to disrupt this circuit, right? So either we can clip this wire or we can make a barrier here, right? Which is typically what's there, but then you get the salt bridge that lets it travel from one end to the other. But those are the things we want to do to get a voltmeter reading of 0. So if we look at these choices, for instance, D just says to replace this with this. Now, and they're the same concentrations. I don't think that'll do anything because this will break apart into the sulfate ion, and this will also break apart into the sulfate ion. And the sodium is a spectator, not going to do anything. Magnesium is going to be a spectator, not going to do anything. So I don't think that will affect it. C, connecting it with a platinum wire. So that's just replacing a platinum wire, replacing a copper wire with a platinum wire. Uh, they're both still going to be conducting electricity, so it shouldn't really matter what that wire is made of as long as it can conduct electricity. So I don't think it'll be that. B, replace this uh, the peroxy disulfate salt here with FeCl3. Now, this is a little better because if we get rid of the S2O8, which is the thing that we need to close the circuit, that's not going to be good. But the FeCl3 actually can help that redox reaction to happen because this would break apart into Fe3 plus and 3 chloride ions. Chloride ions are going to be spectators, don't really care about those, but the Fe3 plus is essentially, what's here, right? Um, like it still participates in this redox reaction. So I think B could still work, meaning like it won't make a difference. Um, but I think it would be A because if we just completely remove this, we remove that. There's nothing that will turn the Fe2 plus back into or to turn it into the Fe3 plus. So I think that is what would close that not close the circuit. It would make it so that the circuit will not be closed.
Wait, but if you remove Na2 peroxidized sulfate, like B, wouldn't that also, screw it up? Because you're not going to have S2O8 2 minus. So then Fe2 plus can't reduce anything.
You're talking about which choice here?
So choice B, if you remove Na2S2O8,
Mhm.
you're not going to have S2O8 2 minus. So
Correct.
Fe2 plus won't be able to reduce anything.
So we wanted that to turn Fe2 plus into Fe3 plus, but if we don't have that and we replaced it with FeCl3, that would create the Fe3 plus.
Oh, okay, I got you. So we won't even need that reaction to happen.
Correct. Yeah.
Because we just get it Fe3 plus right off the bat.
Yep.
Okay.
So, um, yeah. So, yeah.
Okay. And then if you don't add Na2S2O8,
hmm
that's yeah, I guess the reaction just won't be able to happen at all.
Yep. Because unlike B, where we replaced it with something that can still make that redox reaction happen, it seems like we don't have, anything to replace it with. So that'll make it so that we don't have a closed circuit.
Okay. Okay. Next is this.
The migration of sodium and sulfate ions in an electrochemical cell as it operates. So the sulfate ion can go here. Okay. So Um, okay, the Fe electrode was the one that got oxidized, meaning
Yeah,
it,
and mhm.
it loses electrons. So I think that would wait, losing losing electrons, giving it a positive charge. I mean, I'm guessing. Let me see what D looks like. I mean, I'm probably leaning towards C, but
Mhm. That C is right.
yeah. So yeah, so the iron one is where, yeah, oxidation happens. It's the anode, which means it's losing electrons. So if it's losing electrons, it's going to turn into a negative value. Whoops, into a sorry, positive value. So the negative anion is going to be attracted to that, and this side gains electrons. So the sodium positive electron will be drawn towards that. That's also what happens in a salt bridge. Yeah.
Mhm. So
Mhm.
because electrons move from the anode to the cathode. So here it's so this is this a electrolytic cell or what?
Oh, it's electrochemical still. So like you want to kind of link like, like Leo, Gur, but also um An-ox.
Yeah, red cat. But
Yep.
in a electrolytic cell, isn't the anode positive?
hmm
So this would be an electrolytic cell, no?
Oh, no. The so an electrolytic cell, like they would have to draw something connected to an external source of electricity because so a galvanic cell, same thing as electrochemical, same thing as a concentration, same thing as a voltaic. That's how a regular battery works. Just think of a regular battery, right? Regular battery has a charge separation and like like this, like a lithium ion battery just has a lot more of the lithium positive at this top part, and there's like a barrier here, and a lot of and a small amount of it on the bottom part, which makes that bottom part negative. And if I just connected a wire from that negative end to the positive end, naturally, spontaneously, electrons will move from that negative end to the positive end,
Hmm.
right? And this will happen until enough electrons have reached the positive end to give that positive charge and now a charge of zero, and enough electrons have left the negative end to give that end a zero. And this is equilibrium. And this is when you have a dead battery. So what do you do when you have a dead battery? You plug it in to like an outlet or
Hmm.
plug it into another battery, right? But the battery itself is, think about it like this, a battery or electrochemical cell converts chemical energy into electrical energy. So this chemical gradients, separation of charge, if you connect with a current carrying wire, it converts it into that electrical energy. But when it's dead, right, you connect the entire thing to an external source of electricity. And what that does is it forces this to run backwards. So that means, so once you do this, momentarily, instead of a charge of zero here, it'll be positive, and instead of the zero here, it'll be negative because you're running this electrochemical cell backwards. So electrons are now coming from this external electric source and going to that bottom end, which is now positive simply because you're forcing it to run backwards. And if you force it to run backwards and you want electrons to accumulate at that anode must be positive. Okay. And you do this until you recreate the charge separation that you started off with the positive end being the cathode and the negative end being the anode. That's when you have a fully charged battery, and then you can use that. So a battery uses chemical energy to turn it into electrical energy, and an electrolytic cell uses electrical energy to convert it back into chemical energy.
A battery can be both.
So the battery is going to be the electrochemical cell or voltaic cell or concentration cell. So all of that just means you're using you start off with a chemical gradient, which you can think of as chemical energy, but you're converting it into electrical. But when a battery is at equilibrium, meaning no charge separation, uh, it's dead. It's a dead battery. And to charge it, you plug it into an external source of electricity. And once you do that and you run the electrochemical cell backwards, that's your electrolytic cell. And you're trying to recreate that charge separation in an electrolytic cell.
So a battery turns chemical energy into electrical energy. A battery is a concentration cell or an electrochemical cell or a voltaic cell or a galvanic cell.
Mhm.
When the battery dies, you plug it into an external power source and run it backwards.
Yeah, you force it to run backwards because you are recreating that charge separation.
Mm.
It's kind of like,
But
yeah.
the thing is, what do you then classify? What do you, so what, you classify the whole contraption as an electrolytic cell? Like the battery and the external power source together are now classified as an electrolytic cell, or?
Yeah.
Okay.
Just like this entire monstrosity is electrochemical cell. Like it, I usually when I describe it as a battery like this because it's more intuitive. You never see anything like this in real life, but it's theoretically the same thing. Because it's still based on, I mean, it's still based on ultimately redox.
Mhm.
So, but um I thought of like a biological example, which is the electron transport chain. Because when you have in our electron transport chain, like kind of like this, um, like these circles are the things in the electron transport chain, and like H plus gets pumped into this intermembrane space, right? And so
Mhm.
you have a high concentration of H plus here and in the intermembrane space, and in the mitochondrial matrix, you have a low H plus concentration. So there's a concentration gradient there, right? And then when it comes back down its gradient through like the ATP synthase, it can convert that electrical energy now instead of into electrical energy, it converts it into the energy of making the ATP.
Mhm.
And like if you made this membrane permeable, because they like to ask questions like that, like what if you made this intermembrane intermitochondrial membrane permeable, then the high H plus from the outside will just go back inwards. It doesn't have to go through the ATP synthase. It can just go through like this, and that would dissipate the gradient, aka giving you like a dead battery. But yeah, key thing would be that electrochemical cell uh, involves it must have a chemical gradient, aka a charge separation that you can convert and harness as electrical energy until that chemical gradient reaches equilibrium, which means same amount on the inside, on the outside, in one beaker, on the other beaker, etc. And to recreate that charge separation, you can then just plug it into an external battery or electrical source, and you force that reaction to run backwards to recreate that charge separation.
Okay, but so the other thing I don't understand, which is the last question, because I mean we're technically over time already, but like here, while we're on the topic, I want to ask you this: why is the concentrate like from I don't what I don't understand is um from which side gets bigger, like the metal, it gets bigger, and on the side where the electrode gets bigger, um is the concentration higher on that side or in the other side? Do you know what I'm saying?
Let me see. So, let me see the question. Okay. Concentration cell using, okay. Good question. Okay. So, because yeah, typically you'll have two different things and you just go by whichever thing has the higher reduction potential will be the thing that gets reduced. The other thing will be oxidized. But in a concentration cell, you just have one thing. And so the side that has, so like in the lithium ion example that I gave, the positive end is going to be the side that has more of that positive cation, and the negative end will have less of it. So here, so it's in N a nickel 2 plus of different concentrations. And so the plus part, um, like, so it's just like what I drew here, but this is just a high concentration of that. And this is negative because it's just a low concentration of that. And electrons go from the low concentration region of that cation to the high concentration region. And the low concentration region would be your anode, and the high concentration region will be your cathode. And so the electrons will go from the low concentration region, the anode, to the high concentration region, the cathode, since you're dealing with cations.
Um, I always thought that like I guess this is magnetic field, so it's or electric field because it's different. But I always thought that positive goes to the negative, like the arrows.
Oh,
That's different. Is that different?
yeah. So you're right. Current, current was discovered before electrons. So current is, imagine electrons being positively charged. So current is opposite the direction of electron flow.
Hmm.
That can be pretty annoying. Um, so what I'm drawing are like throughout, like everything today that I've drawn has been electron flow. Okay. Current
Yeah.
is going to be the opposite of electron flow. So electron flow will be from anode to cathode. Current will be from cathode to anode.
Okay.
So, but you know, think,
So
yeah.
here's the thing with basically like what I was asking is how do I know which side gains like the mass?
When you say the mass, oh, I see what you're saying. Yeah. So here in this diagram, they're telling us the direction of the electron flow, right?
Yeah.
If they didn't, there's still many ways you can find out. Like they can give you the cell potential for the zinc half reaction, the cell potential for the copper half reaction, and whichever one has a higher reduction half reaction will be the one that gets reduced, which means it'll gain electrons. So here, since they're telling us that electrons are being lost at zinc, zinc is where we have oxidation. Copper is where electrons are going, which means that's where we have reduction. And then anox, red cat. So zinc is where the anode is, copper is what the cathode is. Now, to answer your question about which is the side that's gaining mass, like aka gaining so okay. So the zinc strip is solid, the copper strip is solid. Now the zinc you can see in this picture is losing solid, right?
Hmm.
That means that zinc solid is getting turned into zinc ion at that at for that. But the copper one has solid zinc attached to it, right? So that must mean that copper ion is getting oxidized to solid copper, and that's what we see, why we see more solid copper on the solid copper on the copper cathode. So like this zinc solid here. Oh yeah, here, the zinc solid, that's what this is. That's what this strip is, right? And at I think that's a mistake here. That should be zinc 2 plus. So
Hmm.
so this zinc solid turns into this zinc 2 plus, which is this half reaction. And if you look, is oxidation state 0, oxidation state 2 plus. So that means that it's losing electrons, right?
Hmm.
So so the solid zinc gets oxidized, loses electrons to form zinc cation, zinc 2 plus, right? So that's why this zinc, oh yeah, and then this is a pretty good picture. So you can see like the solid zinc here, and you can see like here, a solid zinc gets lost as this zinc 2 plus cation,
Yeah.
right? And then on for the copper, right? Electrons are reaching the copper, which will, as that progresses, right? Because I know that this zinc is negative, they're telling us, and this copper is positive. But as it progresses, right? The and as the electrons accumulate on the solid copper, it starts to take on more of a negative charge, and we can see that here with these negatives. And that means that it's going to be, getting reduced, right? So these copper cations here get team up with these electrons coming in to form solid copper, right? That
Yeah.
right, and that's what starts to accumulate on that copper cathode. So you see that the zinc cathode it's like solid zinc, but it has these holes in it because some zinc solid atoms are getting oxidized uh, into zinc cation, whereas the copper cathode it has copper cations that get reduced into solid copper. So that's why we have that on the right.
Okay. And so here, the concentration of nickel is higher at the cathode.
So yeah, so this is not going to involve any, since we're dealing with one material, it's not going to be dealing with like plating of some sort. Because that other diagram that you have involves some plating, which is something that electrolytic cells can do also, but with just one thing. But here, since it's just nickel that we're looking at, we can kind of just do it that way. Uh, and so yeah, so here they're just telling us um, that yeah, you have nickel at the anode, nickel at the cathode, and nickel 2 plus. And so we just have to go by our definitions of anode oxidation. So electrons are traveling this way, and if it's losing the electrons, it will be like nickel solid yields nickel 2 plus 2 electrons. That's what's happening at this anode. At the cathode, you have um, nickel. It could be like nickel 2 plus. Well, you know what? If they asked you like what these electrodes would look like after this, it would look similar to like that other picture. Like at the anode, you'd probably have a really like porous kind of used up thing here of nickel solid, and then at the cathode, you'd probably have some, additional nickel solid on that part. So you could still see something like that other picture.
Wait, so you're saying even if it's the same material, it still might gain mass?
Correct, yeah. Because ultimately on the left here, you are getting you are making less solid nickel. And at the cathode here, you're taking the nickel cation and turning that into nickel solid. So it could look like this, what I drew on the left there, yeah.
Okay.
But typically, like, I guess, since it's called a concentration cell, they're going to be likely asking you more about just the relative concentrations of these stuff. Um, yeah.
Okay. All
Mhm.
right. We can stop there. Two things quickly. First of all, what the video you uploaded today
Mhm.
onto your YouTube,
Yep.
do you know what date that was? Oh wait, never mind.
Uh, I can
It's June 7th, I think, right? Or July 7th?
let me see.
Yeah, it's July 7th.
Okay. Was it Bootcamp 4?
Yeah.
Yeah. That's the 7th.
Okay.
And then I'm gonna have one for the 16th. That should be Bootcamp 5.
Okay, so I mean by like November, all the videos from the summer should be up.
Yeah, basically the final video, not including this one today, is going to be the one from August 8th. I think that was the one that
Mm-hmm.
called I think I uploaded that one already for you, I think.
You did. Thank you.
Yep.
So you're saying the August 8th one, like all of them, including August 8th, will be up by when do you think? Just wondering.
So, um, by I guess like this time next week.
Oh, okay. Okay. Perfect. And did you do any research into like what are the best full lengths in terms of company?
Oh yeah. So I yeah, I guess I still have to like kind of do that. Um,
Mm-hmm.
so but I'm looking at so like because there's the Kaplan one you mentioned. Um, like I think the Jack Westin ones could be good. Um, and did you mention?
Okay. The Princeton Review is what I was wondering about also.
Got it. Yeah, I generally have like the Kaplan or Princeton Review ones less because they're kind of like the from like companies that have been doing this for a really long time, whereas like a lot of these newer kind of companies like Blueprint, Altius, Jack Weston and stuff, like it seems like they could have a better kind of hand on the pulse of whatever I
Yeah. Yeah.
can see, but I would have to look into that more. Uh,
Mm-hmm.
but yeah, what I do know for certain is just of course like the Blueprint and Altius ones. Um, but yeah, I can
Okay.
kind of look more into those because you said like your friends really liked Kaplan.
Yeah, I mean, I had like two friends that like Kaplan a lot, but
Mhm.
um, yeah,
Got
that's what they said. But it varies from person to person. Some people, don't like Kaplan, from what I've seen, so on
got
forums.
Yeah. So yeah, I'll definitely have, more information on that, like, once we get to that point of taking tests, like
Yeah.
I'm gonna have, enough tests. Um, so yeah, I would say to for today, um, like I guess like the approaches that we took to going over stuff, but I would say to also look up some stuff on electrolytic cells. Um, because there's electrolysis there, there's or charging, and there's also electroplating. And I think electroplating will kind of help give you an idea of stuff that's similar to the stuff that we did today. Like one thing getting